we demonstrated that we have efficacy in mice. Now we demonstrated that we have efficacy in dogs. What about human? And how can we extrapolate this data from this dog study into human use? And that will be very important for us.
Hello, you're watching Proactive. I'm joined by Dr. Eric Leire, the CEO of Genflow Biosciences. Eric, good to speak with you. You're presenting your SLAB study data at the Animal Longevity Summit in October. In simple terms, what is centenarian SRT6 or 36 gene therapy doing, and why are aged beagles a really good real-world test?
Yes, Stefan, we're very pleased that Genflow has been accepted to present at the result from our aged dogs clinical trial at the Animal Longevity Summit in Toronto. It's a very good fit for us. It's perfect fit, in fact, with longevity animals. That's perfect, but also the audience is very high quality. More importantly for us, the acceptance by an independent scientific meeting is a meaningful external checkpoint. It means that the field consider our data and methodology rigorous enough to put under scrutiny. You can see that as an important step in establishing credibility in the data set. Now, the Longevity Summit will be on October 1st and 2nd. And I want to be clear that the October date reflects the summit calendar, not the pace of our work. There is no delay in our trial or in our business development effort. The timing of the conference slot tells you nothing about the timing of either. Matter of fact, an October public disclosure of data is a safe timing. As a pre-commercial biotech, we're deliberate about what we disclose and, more importantly, when we disclose in order to protect our intellectual property. You know that premature public disclosure can jeopardize the patent right. So what I would like to convey is that we remain in discussion with animal health companies, but under confidential agreement, and we continue to analyze our data collected in the clinical trial, and there's absolutely no delay. This is great that we present in October. It does not prevent us to continue our work with the clinical trial and the business development.
So when should investors be watching for from Genflow up to that clinical trial in October?
There will be the presentation of the data, both sarcopenia and longevity. This conference is also attended by a lot of scientists. And there will be some extrapolation from the work from the dog to the human. You remember that we have also a Sarcopenia human program. And we demonstrated that we have efficacy in mice. Now we demonstrated that we have efficacy in dogs. What about human? And how can we extrapolate this data from this dog study into human use? And that will be very important for us. The quality of the scientists that will be attending this presentation, who will be challenging, offering some suggestions to improve the data and use this data for human indication, is very important for us.
Eric, your chairman is also on a panel this week discussing where RNA therapeutics are heading. Where does Genflow's approach fit into that?
Yes, definitely we move to mRNA LNPs technology, non-viral vectors rather than AAVs. It resolves the main issue of repeated dose. AAVs are interesting in terms of expression, and we had some good results with AAVs, but there's an issue of immunogenicity that prevent to repeat the dose. And when you target tissues that divide, it's very important to repeat the dose to, again, provide the gene of interest to the new cells that come from divisions. So this move to mRNA LNPs has been very clear in all our programs. We have disclosed collaboration with Aquitas. We are working very hard to move our MASH program to transition it to mRNA LNPs. So the presence of our chairman as a speaker on mRNA LNPs make a lot of sense at Bio in San Diego this week.
Well, lots on the boil at the moment. Eric, I hope you'll continue to keep us posted on your progress.
Thank you very much.
That's Dr. Eric Leire, the CEO of Genflow Biosciences.